everyone. So I will be presenting the practical one, which is calculating and describing the statistic, which is the mean, the variance, and also the standard deviation for the leaves. And next is to understanding the concept of correlation, and also determining the relationship between two variables, which is the width and the length of the leaf. So as we can see, we did collect data from 4 sets of leaves by Iqmal, Arif Tarmudi, Arif Hazik, and also Farish. And all the data, all the standard deviation, all the variance, and also means are completely different from each other. So firstly, leaves do not grow indefinitely. At a certain point, they reach a given size and then stop growing. There's this one research from a Danish researcher at Harvard University has found out what determines the limit of how large leaves can grow. It turns out that the height of the leaves it depends. The width and the length of the leaves, it depends on the height of the tree, which is the height of the tree is the determining factor. So the shape of the tree's leaf are a response to the tree species for long-term ecological and also evolutionary history. And ecosystems limiting factors may also modify the finished form of shape of tree leaves. Understanding the logic behind the varied forms of leaves is facilitated by a firm grasp of the precise function of the leaf, which is firstly the leaf must capture enough sunlight for photosynthesis. Second, a leaf must take action for carbon dioxide, which is through the stomata, which can open during the day and night. And yeah, that is all for the discussion. Why is it so different, even though it's from the same species of the leaf? So on the second post-lab question, what does it mean by variance? So variance is defined as a value of squared from mean over the total value, or we say as the squared deviation of a random variable from its mean. Informally, it measures how far a set of numbers are spread out from their average value. So the meaning of the statement, the leaves have different measurement of width and length. About the measurement of width and length on different leaves have different values, since the leaves come from a different part of the plant, which means that the part of the leaves we get was from 3 different parts of the stem, which is from the upper part, the lower part, and also the middle part. From that sentence, we can say that the leaves have different measurement of width and length due to the location of the leaves that were taken from different parts of the stem. So next is standard deviation, is defined as a measure of the measurement of the amount of variation or dispersion or of a set of values. It also is used to show how the measurements for a group of data are spread out from the average or mean. Standard deviation is also divided into 2, which is low and high standard deviation. Low standard deviation that indicates the value tend to close to the mean of the set, and a high standard deviation indicates that the value are spread out over a wider range of values. So the third post-lab question, why is it necessary? So it is necessary to have a different sets of measurement, from measurement of the leaf width and length, to to get a result that shows the plant contains the same characteristic or not. So it is also necessary in order to obtain the valid data and results where we can have an average measurement of leaves instead of only use a few leaves.
Let's move on to the practical 2. We have done the 2 quadrat. The quadrat A, we have a lot of the various tree. For example, we have the Garcinia acronividis, or we call the asam gelugo, or the Shoria ovalis, or we call it the meranti kepung. Each of the organism or individual is one only. So the density of the organism, we use the formula number of individual over area of the quadrat, so we can get 1/25 meter square, or we can simplify it with the 0.04 m square. Meanwhile, for the quadrat B, we have the 5 type of species name. For example, we have the Syzygium species, or we call it Clyde, and Hippia odorata, or we call it the siput, pokok siput jantan. So it same goes with the quadrat A, with the total number of the individual is 1 only. So the density is same with the quadrat A, which are 1/25 m square, or we can simplify it with 0.04 m square. For the question 1, define the term quadrat in practice. Okay, in this battery, a quadrat can be defined as the frame that is typically square with various size, and it used to isolate the standard unit of the space for the study of the subject distribution over a vast area. For instance, contemporary quadrat can be irregular, round, or rectangle. The quadrat is useful for the collecting sample for aquatic species, slow-moving animal, and plant that reside in the particular quadrat. In this practice, we use quadrat to study the distribution of the tree species in the specific location. Okay, for the question 2, what are the 2 basic requirements of the quadrat technique? Okay, in my opinion, the 2 basic requirements for all the quadrat technique is the area of the particular area of study is known, so that the density of the organism can be determined directly. The other one is the organism that are going to be studied must be immobile during the counting process, so that the count can be precise and none will be missed. So the last question for our lab is, what will be the advantage and disadvantage of using a larger smaller quadrat to survey the sedentary organism? Sedentary organism is the organism that inactive, for such as the basil or the snail, and or maybe we can assume the organism that's slow. The advantage of using larger quadrat is, if we use the bigger, we can get a lot of data, but however it can cause a lot of error during the calculation. But same goes to the smaller one. The advantage of the smaller is it can reduce error and fault calculation, but the disadvantage is it has a small area, so we can get a small quantity in organism.
Assalamualaikum warahmatullahi wabarakatuh. Okay, my name is Muhammad Ikmal Arifin Jerry, and now I'm going to talk about the field trip, our field trip to the Putrajaya Wetlands Park in Putrajaya. Alright, so we went there on the evening side in the Friday after the Friday prayer. Alright, so what is Putrajaya Wetlands Park? Alright, so Putrajaya Wetlands Park is a park that is located in Putrajaya, of course, in Malaysia, which is the largest constructed freshwater wetlands in the tropics. It is the first man-made wetland in Malaysia, which includes a wetland park and the wetland areas. The Putrajaya Wetlands Park consists of 24 wetland cells, wetlands park, and other wetlands areas. So it is a very beautiful park to visit, and it's very worth it to visit. Alright, so let me tell you about the experience that me and my class when we went there for our field trip. Let's go! Alright, so when we arrived there using our designated buses, alright, so we were given a briefing and a short introduction to what Putrajaya Wetland Park is. The brief was given by the staff there, and the information were given including the purposes, the areas covered, and also the specialty of the Putrajaya Wetlands Park. Alright, so after we were shortly briefed, then we were divided into several groups consist of 4 peoples for each group. So as for my group, includes me, Muhammad Kamal Arif, and Arif Darmudi, Arif Haziq, and the other one is Farish. Alright, so 4 people for each group. Alright, so then each group was given a file which contains some paper forms and a scissor and a cellophane tape for the next activity. Alright, so then we were required to find the appointed plants that is appointed in the paper form. Then we must cut a part of the plant. We must find the plant and we must cut a part of the plant and stick it to the paper form using the cellophane tape. So it's very interesting activity to do in the wetland park, right? So the group with the most correct plants collected according to the designated names in the paper form will win. Alright? So for example of the plants that we are required to find them, such as water lily or nymphea, its scientific name, woolly sage, Carex pellita, soft stem bulrush, Schonoplectus tabernaemontani, and the last one is cogon grass, which is Imperatus cylindrica, or in Malay we call it as lalang. So these are a few plants that we are required to search in the wetlands, and we are required to stick it in the paper form. Alright, so then we start our little journey in the Putrajaya Wetlands Park, which is the most interesting part. And here are some of the footage recorded during our little journey. Let's go!
Okay, lagi 3 guys.
Jom.
Tak aku.
Ada yang tak tak terus lah.
Bangau.
Yang siapa ni?
Ya, aku cari ubi kurun ni.
Di depan banyak lagi.
Buatlah video rakam-rakam sikit ke?
Cepat rekod je.
Alah.
Mari cepat.
Alright, so in the end of the activity, we were required to regroup back at the end of the Putrajaya Wetland Park, and we have to give some conclusions on what have we obtained or received from the activity that we did there. Alright, so that's all for the activity, and then we went back to Item Shala in the evening using the designated bus like how we came. So here I would like to express my thanks to our lovely Dr. Faiza Fardi for bringing us here together with our classmates. We had a lot of fun doing activity there, and we had learned a lot of things such as the plants, the purifying plants, right? So I hope there will be more field trip together like this in the future. Thank you, Dr. Faiza. That's all from me. Thank you.